Okay. Well, thanks, Sergey, for the opportunity to present here at this ML Virtual Summer School. So up till now, you've heard a little bit about some basic machine learning, some basic image processing, spectral processing, and so forth. But today we're going to discuss kind of the meat of machine learning, which is what happens underneath the hood. And that is related to code and data infrastructure. And I think this is a part that often gets overlooked in many ML courses. And so that's kind of one of the reasons why we wanted to have this lecture today, to explain to you what is the importance of code and data infrastructure, what is the importance of reproducibility, how do we get there, and so forth. And so we're going to use our solution, which is the PyCroscopy ecosystem of packages, to give you an example. You don't have to necessarily use this one, but the point remains that data and code infrastructure is kind of critical for any machine learning workflow. So I guess if you have any questions, you can interrupt me if necessary. But we can also discuss at the end of this particular presentation as we go through the notebooks themselves, because I think a lot of the questions will be answered in those notebooks and in the exercises that we do there. So let me start off with sort of a question about why do we bother with infrastructure, right? So generally, people take infrastructure for granted, right? So people don't notice that a road is cracked and got potholes until they drive over it many times and realize that, oh, the conditions of the road are deteriorating. And to a large extent, this is also true of code infrastructure, right? So if you keep continuing to use a package, but you don't update the package, you will notice that eventually it becomes out of date and it no longer works. And then none of your code actually works, and then your ML can no longer proceed. So it's really important to have a good handle of what code and data infrastructure you have within your particular ecosystem, and know how to use it, and know what are the weak points, and know how to overcome those weak points. And really, this is because almost all the machine learning relies on access to sort of good quality data, as well as good quality compute environments, right? So where can you perform the computations required for machine learning? In most laboratories, right, this data is acquired by different instruments. It is often in different formats. It often doesn't have the metadata involved. It is neither findable nor accessible in an easy way. So if you work in any laboratory, generally, you know that the way that data is shared is via USB drives or Dropbox folders. Things are not particularly labeled, or if they are, they really require you to go back to the individual who collected the data to understand better what this data is actually about. And so there's really a lack of metadata that is required for machine learning purposes, right? If you're doing supervised machine learning, or even for that matter, unsupervised machine learning, you really need to know what kind of data goes into your model. And without having this data in a standardized fashion, it can be really challenging to perform any realistic machine learning outside of some toy examples, right? And I would say that this is probably also why most machine learning material science has to date occurred in the simulation realm, because it's much easier to standardize simulations than it is to standardize experiments themselves, because of the different varieties of experiments that you have and the different kinds of data that comes from any individual experiment. It can be more challenging and more heterogeneous. And so we have come up with methods to be able to standardize that, at least in the microscopy realm. And so that's partly what I'm going to talk about today. So, in many cases, machine learning in science is basically impossible in experimental domains without the necessary investments in data standardization and storage, right? So if you don't have your data centralized in some kind of repository, it can be very challenging to perform machine learning, because then it is up to the individual to go and try to accumulate the required data. And this is very time consuming, much more time consuming than any algorithmic development that might have to take place. And similarly, reproducibility of workflows relies on strongly tested code bases. So what do I mean by this? I mean that, if you perform machine learning by just sharing a bunch of scripts around, like that can work at the very low level, right? So if you have 2 or 3 people, collaborators, and you just have some scripts that are working, then you can share those and they can be useful for a short period of time. But as anybody knows who's been in this business for long enough, you know that that doesn't work after a couple of years. Generally, those codes become defunct and no longer workable. And so what you need is, if you want to be able to reproduce your workflows over multiple years, you need strongly tested code bases, right? Not just one-off scripts. And so this is where version control and packages and software development really comes into the picture. And as I mentioned before, PyCroscopy, which is sort of a play on Python and microscopy, is sort of our method to alleviate this particular problem. So as I've been mentioning here, really all of machine learning is built upon this sort of a tip of the iceberg kind of pyramid kind of structure, right? So if you think of machine learning, we think of things at the top here, right? Algorithms, like how can we apply our algorithms to scientific data? How can we incorporate simulations and so forth, right? But really all of that sits on top of, a larger brick of scientific data, and really underneath that is hardware and software infrastructure that enables all of this computing to take place, right? So without the hardware and software infrastructure that is sort of underneath the ocean that people don't really realize and people just take for granted, you're not going to be able to do any of the machine learning that we really want. And particularly in the context of autonomous experiments in labs, you may be able to get away with not worrying so much about hardware and software infrastructure for one-off machine learning projects, but in an actual working laboratory where you are trying to incorporate machine learning workflows into experiments, without this, you are not going to succeed, right? And so as anybody in industry will also tell you, this is the main part. This is 90% of machine learning is sort of down here. And so this is why we're discussing this today. So what is PyCroscopy? So PyCroscopy is a sort of an ecosystem of Python packages for microscopy data ingestion, analytics, and visualization. And so we sort of combine multiple different packages together within this ecosystem. There is also a Python package called PyCroscopy, which is a general purpose package for microscopy imaging and spectroscopic data analysis. It does things such as image cleaning, matrix factorization, tensor factorization, and so forth. These are really sort of general purpose, non-domain specific kind of tasks. But we sort of split up the actual ecosystem into multiple different packages dependent on whether we are dealing with file input outputs, whether we are dealing with things that need to be written to file or just kept in memory, and then as well as a bunch of specific domain-specific applications such as BGLib, which is band excitation and G-mode spectroscopy, Atom AI, which is made by Maxim Zyatinov, as well as our collaborators. And so here, people like Ayana Ghosh and so forth at Oak Ridge really contributed significantly to Atom AI. But this is Maxim's package where we can enable workflows involving deep learning for particularly atomically resolved images, but also other tools such as PyTemLib, which is made by our collaborator here, Gerd Duscher, who's also been a lecturer here, which enables simulation and registration and other visualization and analysis of TEM data. And so we sort of split up the Python ecosystem of packages into these different kinds of individual applications. And the most important one probably for you all at this point is the Sci-Fi Readers package, because this is how you get data in and out of the ecosystem. And so what do I mean by that? What I mean by that is, let's say that you get some data on the stem, you have a raw data file. And so what we want to do is we want to convert this data file into a standardized format that can be read throughout the PyCroscopy ecosystem of packages. And so you take this raw data file, and so then you read it using Sci-Fi Readers. And then what Sci-Fi Readers does is it reads this raw data file and puts it into an open source format, which we call a SydPy dataset object. So this is represented by this D here. And so what happens is that once you have the SydPy dataset object, you can pass this around through different packages within our ecosystem. You can perform analysis, and then you can then output that analysis to a HDF5 file using either NSYD or USYD formats, which I will get to later. But this is the idea. The idea is that when you have multiple different data sets from different sources, you can then standardize all of this. These data set objects contain all the metadata that is required for you to do your analysis and to maintain all of the metadata that was captured during the experiment. And it is also a fully open, standardized and tested platform. And so this is what we are using in order to standardize our microscopy acquisition. So why do we need standardization of formats, right? So here's the standardization. When you look at an individual laboratory, you might have multiple different characterization tools, right? So this workshop is about STEM, but it's also true for things like atomic force microscopy. You can have AFM platforms with things like infrared spectroscopy. You can have them with Raman spectroscopy, or you can have micro Raman instruments. it really doesn't matter. Basically, there's a large variety of scientific characterization tools that exist in any laboratory, and unfortunately, all of these have their own formats, right? So STEM data might be produced to you in TM3. It may be produced in other formats. Some of these are produced in sort of MATLAB.mat files, and some of them have their own sort of proprietary formats here. And so the question is, what do we do with all this, right? So typically what people do is that they go ahead and collect some individual data from these places. They might save them as NumPy data sets and then import them into a notebook, and then add the metadata themselves. And so this is really not a workable solution in the long run because it requires too much user effort and people will not go through that effort. But what's a better solution is that if we can save all of this in a regular file format, which maintains all of the provenance as well as the metadata that was captured, then we will be able to standardize data across all of these platforms, regardless of what kind of experiment was performed, right? And if you think about it, this is actually quite a challenging task, right? If you are asking for a single data model to be able to handle all of these different kinds of experiments, then you need to be very careful and considerate about what kind of data model you're going to use, right? So typically, most of these formats are written with some specific modality in mind, but what we need is a more general purpose microscopy data file format. Right. And so the other problem here is that most of these particular modalities or most of these particular instruments have their own analysis packages, which they use. And so this is also not particularly great for code sharing, right? So what we would like to do is we'd like to have an ecosystem which works in the most commonly used software, open source software platform, and that's Python. And so what we want to do is we want to have a format that we can run Python analytics on, but also that is able to ingest data from all of these different kinds of instruments. So, we cannot sort of share code efficiently if we don't standardize these acquisitions, right? Because of different programming languages, sort of highly instrument-specific code, as I mentioned. And then the other thing that I did not mention is that a lot of the times, these require, costly or licensed versions of software. MATLAB is a great example, but there are numerous others. This is okay for small labs, right, to sort of pay for a site license and acquire some of this software, particularly for universities. But outside of university settings and in any industry, you will find that these site licenses are extremely costly. And they also really prohibit you from sharing code with a wider user base. And so we believe that this should be mostly avoided. Also, sharing code by email is a very common way, and I do it all the time, but it is not necessarily the best way, and it is certainly not a reproducible way, or it's not a scalable solution, right? So what we want is a centralized repository that is written in a free and open source platform. And so for us, that's really Python. So what is our solution, right? Our solution is basically have an automated and a standardized data model that can represent data that is captured across multiple different instruments, sort of have save it into a self-described model in an open and friendly file format, and then have these centralized repositories for data processing analysis. And then you could also use this sort of interactive visualization and analysis and storage on the cloud once you have all of the previous steps completed. Okay, so I've talked about, what is our solution, but I never went into specifics. So I'm going to talk about two particular specific data models that we're using. And so the first one is called the Universal Spectroscopic and Imaging Data Format, or USID file format, or data model as we call it. And so this is a data model that really allows you to store any kind of spectroscopic measurement that you might perform in a microscope, right? So, and this is true for any dimension that you can think of, right? So what do I mean by that? What I mean by that is, let's say that you have 5 or 6 dimensional data sets. You can store it as a USID file. And the way that we do that is that we effectively reduce our data to 2 dimensions, right? And so what are those 2 dimensions? Those 2 dimensions are position which goes along individual rows, and then a time or spectral axis, which goes along the columns. And so what we do is we take any data that's given to us and we break it up into these two dimensions, right? So if it's a microscopy data, typically either it's spectra at different positions or it's images. And so we can represent any kind of spectroscopy that is performed using a microscope using this particular data model. Now this, you might ask, so if the data is already in sort of 3D or 4D formats, why do we then want to reduce it to 2 dimensions? And the reason for that is because unfortunately, although some data can be stored in sort of n-dimensional formats, not all data can be represented in n-dimensional formats. And so for those situations, you really need to use a 2D format in order to be able to represent that data. Let me give you an example. Let's say that, for example, I am measuring a spectra at a particular position as a function of some other variable, let's say voltage, right? And so let's say that I have a voltage waveform and I vary this voltage waveform across time, and I'm measuring the response as a function of this particular voltage. Now, it's possible that this voltage waveform cannot actually be reduced to an n-dimensional representation, right? So if, for example, I have an on kind of waveform where I'm measuring the response while this voltage is on, and those on steps are, let's say I have 16 time steps, right, for those on portions of the voltage waveform. And then I measure, let's say, 32 time steps where I turn the voltage off after that, right? So unfortunately, because this particular spectroscopic dimension of voltage has an on portion and an off portion, which have 2 different time step lengths, we can't actually then fold it back into an n-dimensional representation, right? So we have to split up the data into an on-field and an off-field representation, which makes things more complicated. And also prevents you from analyzing the whole data set as one. So what we came up with to try and store this kind of data is this sort of 2-dimensional representation, where you simply store all of the spectroscopic values, or the response measured as a function of that spectroscopic variable across these columns, and then all the individual positions are measured and added as rows to this 2-dimensional matrix. Now, when we do this, we also have obviously measurement or analysis parameters. And so these are stored as attributes within this particular file. So we can store all the metadata that is necessary. But also, we also need information about the individual spectroscopic axes themselves, right? And so we have 4 different kinds of ancillary data sets that we call them. These are position indices, position values, spectroscopic indices, and spectroscopic values, right? And so I'm not going to go through these in too much detail, but other than to say that when your spectra are varying across multiple different dimensions, this particular ancillary data sets model allows you to store all of those particular spectra with their representative values of the different dimensions. And so this allows you to reconstruct the full data set using these full ancillary data sets, as well as the sort of main data set here, which is this 2D data set. And so I would say that, although this particular data model is widely used at the CNMS at Oak Ridge, I would say that it is more complicated because of the need for these ancillary data sets. And so if your data is not capable of being stored in an n-dimensional form, then I would say you should use this UCID model. But if you can store your data in n-dimensional form, and that is probably 90% of cases in microscopy, and particularly in STEM, then I would say that this is probably not the particular data model that you want to use, because it is more challenging to use given the need to keep track of 4 ancillary data sets. And so I would say one more thing, which is that if you have point cloud measurements, so let's say that you have your beam and it's positioned at different, let's say that you position it at different atoms or different atomic columns, and you measure the spectra at those particular individual positions, then it is very easy to store these sort of point cloud measurements in this particular model. But if, on the other hand, you have n-dimensional data, such as a hyperspectral EELS data set, where you're just collecting across a grid of points, and you can represent this as a 3-dimensional hyperspectral data set, then this might not be the model for you. Instead, what we choose, I'm just going to sort of skip over this because I don't think it's necessary for this, but I'm happy to go over this at a later date if later on if you want me to. But what we choose to utilize instead, and which is what we're going to work with today in our notebooks, is the so-called NCID model. And so this is the n-dimensional spectral imaging dataset model, which we came up with, which was a successor to UCID. So UCID is really good, and it's really general, and it's really applicable to almost any kind of experiment you can think of in the microscopy realm. However, given the fact that you need to keep track of all these ancillary data sets, it can be quite challenging to use. And so if you have data that can be represented as an n-dimensional model, then you can use this so-called NSID model, which is implemented as a SIDPy dataset object within the SIDPy package in PyCroscopy. And so let's say that you have a dataset where you have an image, which is 128 by 128, and then at each individual pixel in this image, you have a spectra of length, let's call it 31 here, right? So you can imagine this is kind of a cube of data. And so when you look at these particular SIDPy dataset objects, if you just type a SIDPy dataset object into a notebook and press print, you will see this particular representation. And so this is showing you that, in fact, if any of you are familiar with Dask arrays, SIDPy datasets are effectively built on top of Dask arrays. So we take the advantages of Dask arrays, which is that they can be very, very large. And so we can also store dimensional information over Dask arrays. Then what we can do is and also allows us to do parallel processing. What we can do is we can build on top of the advantages of Dask arrays, but really incorporate information about the metadata that is associated with each individual axis, as well as metadata about the type of experiment that was performed, the type of imaging data that we've actually captured. And so the benefits of this model are that, it's easier to understand. If you have an n-dimensional matrix, just go straight in as an n-dimensional matrix in a SIDPy dataset object. It also takes care of plotting, right? So if we take this particular SIDPy dataset object and we just do a .plot command on any SIDPy dataset object that is correctly created, then we can plot that dataset regardless of its dimensionality. And because we leverage Dask, we can easily perform parallel computations. And so it's sort of easy to push to files from SIDPy dataset objects. So these are dataset objects that are sort of in memory, but if we want to then store them as files, it's very easy with a single line of code to then push them to a HDF5 file. And so because of the standardization, it's really useful for data pipelining. So let's say I'm getting data from multiple different instruments. I can convert it into this SIDPy dataset object. I can push it to HDF5 files, and then I can then read via metadata all of the files that I need to for my particular ML task. Right. So what model should you use? Should you use the NSID model or the UCID model? I would say that, in your case, mostly you should use the NSID model unless you have a particular use case for a UCID model where NSID representations do not work. But I would say that for the most part, you will not come across those in STEM. I may be proven wrong, in the future by this, but I think that in general, you can more or less stick to SIDPy dataset objects, which are built on top of the NSID data model. Also, both of these have sort of enable parallel processing capabilities. I would say NSID is a little bit slightly less rigorous about traceability than UCID is, but we're getting there for this particular data model as well. And it has a much easier ease of use than UCID does. Also, we have at Oak Ridge a tool called DataFed. And so once the data is standardized in this way, where we can push it to HDF5 files with all the required metadata, we can then read it using this federated data repository tool. It's called ORNL's DataFed. It is an open source solution. If you want to, you can have a look at it at datafed.ornl.gov. But essentially, this allows us to then search through our data via metadata, and also do interesting things like search through by thumbnails or search for individual parts. Let's say that I want data that was captured on a particular instrument on a particular date, or I want data that is captured on a particular kind of sample. Then I can look through the metadata and then pull up all of the entries associated with that. And that becomes really useful when I want to collect particular kinds of data for my machine learning workflows. All right, so the next thing I'm going to talk about here is sort of code repositories and version control, right? So most of you are familiar with Jupyter Notebooks by now. You've been using them for a number of weeks. You probably are also familiar with some level of version control, or at least the fact that version control exists and there are code repositories available, even if you don't particularly understand them very well. And the reason for that is because basically all of the code that you are using has been pushed to repositories, and those repositories are where you are running your notebooks from, actually. And these repositories also have version control inbuilt into them, right? So why do we do this, right? So as I mentioned before, sharing scripts between users can be workable for immediate needs, but it is neither scalable nor long-lasting. And so for reproducibility, what we really need is we need to have codes that reside in packages that are both documented as well as well-tested. Now, I would say that probably 90% of code that is written in the machine learning world is not going to be in packages that are well documented and well tested. Only a small subset of the codes will be. Now, that's okay in research environments because you only want to sort of invest time in this for the codes that become very useful for your particular group. But I would say that if you develop something that you think is useful, then having a simple script out there where it runs is not likely to be particularly helpful to the community. However, being able to incorporate that particular algorithm into an existing code base or creating your own code base, should there not be an existing package for your needs, would be a much better way in order to be able to get that code out to the largest number of people who can then use it. But there's a few things that have to happen here. So one is obviously that the code has to be well documented. And I would say that with the advent of ChatGPT, code documentation has become a lot easier, and code testing has also become a lot easier. So you can use things like ChatGPT in order to create, or GitHub Copilot in order to create some doc strings in your code. And so if you create a function, you can then immediately, use these tools to help you write the documentation for those particular functions. And they can also, in some cases, write some testing functions as well. But the point is that most of us are not trained in either code documentation or in code testing. And so this is something that we have to learn by ourselves. But I think it's well worth the effort to go through and make sure that code is well documented as well tested, because the worst thing that can happen is that some minor change is applied to your particular code, and then everything stops working, and then you don't remember what was actually changed. And so this is why we need code that's both tested as well as we need version control, so that we can always go back and have a look at all the changes that were made and then move back in time if necessary to be able to see what changes were made and where things broke down. And so what we're going to do today is we're going to go through a version control system called Git. You probably already are familiar with it because the notebooks that you're using here and the lectures are all online on the GitHub repository. But we're going to do some work on Git today. And just very briefly, if I wanted to talk about Git, I probably have to talk for another 3 hours because it's a very involved kind of piece of open source software. But Git is an open source distributed version control system, and it maintains a history of changes that have occurred in the project. So typically on any software project, there's not just one person working on it, there's multiple different people working in parallel on any particular software project. So when people make changes to the project, you need to know what changes were made, who made them, and whether they are breaking anything within your particular code base. And so version control is really important as part of the sort of continuous integration and continuous development environment that software developers like to talk about. And so it allows us to be able to find out what changes were made, allow for updates, as well as reversions to older so-called commits. And so we'll go through some of that in an example soon. So I'm going to leave you with this particular XKCD comic, which is that the Git command line tool is very extensive and very powerful. However, it is can be very confusing. And so I thought that this is a very nice kind of illustration of how confusing it can be. That said, you don't need to necessarily use command line tools for Git. There are lots of software packages out there, including GitHub.com's own client, which allows you to have a graphical user interface to be able to use Git. Also, a lot of your code editors, interactive development environments such as VS Code or PyCharm, they also include version control software, including Git. And so you can run your Git commands without actually having to know any of the Git commands by heart using these tools as well. So I found a nice Git tutorial on Atlassian. If anybody's interested in learning more about Git, I would strongly encourage you to go and have a look at that particular tutorial. Also, there are many YouTube tutorials online where you can have a look at how to use Git and what its basics are. But for now, what we're going to do is we're going to test the basic functionality of Git in an interactive demonstration. You must have a GitHub account, which I assume most of you do. And so what I'd like you to do is to head on over to this particular link. This is my name here, and this is my GitHub account. And so I have a repository called ML Summer Course. And so as I pull this up, please also do that on your particular computer. So I'm just going to stop the sharing right now. And then go on to my GitHub account. Okay, so can everybody see this?
Yes.
Okay, good. All right, so if you go to GitHub and you search anybody's name, you will find the repositories that they have that are public. And so the one that I'm going to do here is I'm going to look at this ML Summer Course one, which is just a quick repository that I made just for the purposes of this particular demonstration. So what we're going to do here is that whenever you see a repository like this, you will see obviously that this one is a public repository. And what's listed up here is that we are on the so-called main branch. So what does that mean? That means that, let's say that you have multiple different people that are working on your particular project, or you want to do some updates on your particular project. Very often you have a so-called main branch, which is, as the name implies, the main branch of your particular package or your particular repository. And what you can do is you can then look at other branches that people have created, right? So very often these are called things like dev, or some other feature that wants to be added to this software package or repository. And so you can find other branches within the repository by clicking on this button. Right now we don't have any other branches, and so there's only the main one. But that's just for your future reference. So what I would like you all to do is I would like you to all fork this particular repository. What does this mean? So what this means is that you all have your own GitHub accounts. When you click on this fork button, what it will do is it will then fork. So I can do this. And so what I can do is I can choose an owner, and I'm going to choose a different name. And so what I can do is I can copy this repository into my own folder, right? So into my own GitHub account. And so since I also own the PyCroscopy account, then I can fork it into here. I'm not going to do that now because I already forked it previously. But once you create this fork, what it will show you is that it will take you to your particular version of this repository. So what all this is doing is it's copying this repository and putting it on your particular account. Okay. So let's go there. Actually, I may as well just do this anyway, and then I can delete this afterwards. So you can give your repository a name. Typically, it's just the same name, but it's okay. Create this fork. Okay, so what do you see now? So what you see now is that right now I'm in PyCroscopy. So this should actually be with your GitHub account. So I'm just going to give people a couple of minutes until they've done that. And let me know if you have done it or you need some more time. Okay, I see Russell has done it. Okay, thanks, Arvind. All right, so what we're going to do now is we're going to make some changes to this code, right? So how do we do that? So what we do is we use the inbuilt editing capabilities that are on github.com. We can just click on one of these files, for example, python_file.py. This is the code. And so what I can do is I can click this particular edit button. So I'm going to say, let's say, okay, so what I'm going to do here is that I have a particular script here, which basically makes some fake data and then tries to plot it and then fit it to a polynomial of second order. So we are going to make a change to this code base by, instead of using the second order polynomial, use a third order polynomial. Really simple, right? So what I'm going to do here is I'm going to comment this out and then add 3 instead of 2 here, and then commit changes. And so what this commit changes will do is that it will ask me to have a description of what I did, right? And so what I'm going to say is I'm going to say updated to include third order polynomial, okay? And so I can either commit this directly to the main branch, in which case the main branch of this particular repository that is in PyCroscopy now, or it should be in your account, will be updated, or you can also create a new branch, but we're not going to use that now. We're just going to commit directly to the main branch. All right, so let's commit these changes. All right, so now if I actually look again at this repository, you'll find that, you know, I made an update here just now. And so there were 4 commits here, but the last commit was by me. And so that commit means that whenever you are ready to make a change, you sort of commit that change. So that's what that means. And so if I look at this file now, you can see that now we have updated this particular file here. Okay, so that's all well and good. That means that you have updated your version of the code base. But typically what you want to do is that when you're ready, you want to actually sync these forks over. So what does that mean? That means that I made a change to this repository and I want the original owner of the repository to accept my change, right? And so how do I do that? Well, the way that I do that is that I can click contribute and then I click open pull request. So what does that mean? What that means is that it's going to request the owner of the original repository. So in this case, it's actually this account here. It's going to ask them to please look at the change that was made and accept that change to the original repository. Okay, so let's click open pull request. And so again, it's just going to show it's going to show me that we are pulling from this particular head repository. In other words, this is the repository where I want to merge my changes from here back to here, right? So and GitHub has already checked that we are able to merge these changes without any conflicts. So if there are some conflicts, which can happen when multiple people are sort of editing different files and different segments of the same file together, which requires some conflict resolution, which is usually done through something called a git merge tool. But we're not going to go through that today, but just for the purposes of this demonstration, we can just click create pull request. And so if I actually look at this now, you see that in fact, I'm back to my original repository, where this pull, if I go to this ML summer course, I will see that there's actually 3 pull requests that are outstanding. What does that mean? Well, there was one that was done, it seems, by a user called Natrium77. And so they have opened a pull request. Let's check out what that is. And so here it says updated for third order polynomial. We can have a look at what's in this pull request by looking at what changes were made. For example, if I click on this, I will be able to see what changes they made. There we go. So if I come back, I can then say, I would like to merge this pull request. So what that means is that I'm going to accept the changes that this user made to my package, right? So I'm going to confirm the merge. So I hope that you can also submit pull requests. Only the owner of the original repository can accept these pull requests, or the people who have moderator status on this particular repository. or so-called maintainer status. So I'm still waiting, and I'll wait on a few others to submit their pull requests. Is there any questions? Is anybody running into any issues? All right, I'll just give them a bit of time. Okay. Okay. All right. So I don't have any pull requests yet, but I'm assuming that that will come in. But in the meanwhile, I think I'm going to hand off now to Maxim, who has a little bit more information about some code and software ecosystems, and particularly how they can connect to computing infrastructure. So I'm going to hand off to Maxim.
Okay. Thank you, Rama. Hello, everyone. Good morning, everyone. Okay, so let me try to share my screen. Let's see if it works. Okay, hopefully you can all see my screen, and let me know if you cannot. Okay, so I wanted to briefly chat about edge/cloud compute framework or ecosystem for autonomous microscopy. So this will be a very, like I have maybe 6 or 7 slides, and this is going to be a very high level overview, because the specifics of each framework or each ecosystems are different from one institution to another. They are different between national labs and universities and industry and so on. So I'll just give you a very general overview. Okay, and so if you think of a typical human-centric workflow, experimental workflow, then you could describe it this way. You have an instrument which it doesn't have to be necessarily an electron microscope. It can be, let's say, scanning tunnel microscope or something else, but basically some instrument that generates images and spectra, right? And then you have a human scientist that who views that data, and based on his or her prior knowledge and quite frankly, most of the time, intuition, decides what to do next, right? Actually, I forgot to draw an arrow here, but it goes from instrument to images and spectra to human scientist, and then a human scientist makes a decision about the next measurement. So the problem is that given the volumes and dimensionality of the data that can be acquired in a matter of seconds at a modern microscope, a human scientist just simply does not have enough time or mental capacity to go through all the data and make a decision informed by the analysis of this entire of all the data volumes, right? So then the question becomes, okay, can we replace a human scientist with AI? And by AI, I put it in scare quotes because right now, these days, the definition of AI is so vague. But the idea here is that it's a kind of a black box that takes this image and spectral data in and outputs allocation of the next measurement, a location in the parameter space. So it can be simply XY coordinate where you want to perform the next measurement, XY coordinate within your sample, or it can be some coordinate in a more sophisticated and high dimensional parameter space, which could include temperature, pressure, and so on. But that's an idea. And the reason we want, again, to use AI is because of its awesome pattern recognition powers. But this awesome pattern recognition powers also require awesome compute powers. And so, unfortunately, this is not something that you can do on a CPU of a computer that comes with a microscope, right? And so you will hear a lot, I think, about this in the future lectures. But a lot of, in many cases, what we are doing is the so-called active learning, where we have an AI model that constantly interacts with the data generation process and learns from it. And so after each new measurement, we update model by retraining it, and then use this updated model to guide our experiments. And so the point is that this step where we essentially do model training is very computationally costly. And again, it cannot really be done on a CPU of a computer that comes with a microscope. And it does require access to state-of-the-art GPUs. Now, I should point out that there is, I don't know if any of you follow me on Twitter, few weeks ago, I pointed out that there is a huge discrepancy when it comes to the hardware development rates. So the huge discrepancy between the hardware development rates for instrumental sciences and for the AI, right? So on the AI side, you see, NVIDIA releasing better and better GPUs, more and more powerful GPUs every single year, sometimes twice a year. On the instrument side, I mean, changes are much slower, maybe like, one change in 10 years or something. And because of that, the things that require access to high performance computing 5 years ago can now be run on a simple video graphic card from NVIDIA, right? The one that actually they develop a video game in, but it's actually more than enough to run many experiments. So so for a lot of stuff that for today we have to send data to external resources, I think in a few years you will just able to purchase a single graphic card from NVIDIA and run everything on it. Okay, but we're not there yet, and because of that, we have came up with a solution where we send data through our internal network to external high-performance computing facility, where so basically you have a microscope in one building, and it doesn't have to be microscope. This is a very general framework developed by our colleagues and our collaborators, Nagirao and Anis, and so like 99% of the work was actually done by them. Anis has this nice paper in eScience where he describes how this was all done. So I'll just give a very general overview, but basically you have your instrument in one building, and then you have your computing facility, which is most likely in a different building. And this computing facility can be anything. It could be, a cluster of CPUs. It can be an actual supercomputer. It can be a bunch of NVIDIA DGX boxes and so on. So it doesn't really matter for the purpose of this presentation. And then basically you have a control channel for remote instrument steering. We have data channel for streaming measurements across different facilities. And this kind of setup allows cross-facility analytics and science workflow orchestration, right? So this is just a very general overview. Again, please, if you're interested in the specifics, if you are into this, networking and infrastructure, please check out Anis's paper in eScience. It was just published recently. So for our specific problem, which is electron microscopy, we basically, so this is the adaptation of that solution to this very specific problem, where the way it works is that in principle, assuming that your sample is already in, and you have, well, actually, yeah, already your sample is in, your Microsoft is obviously turned on, and you can log in from your home in an external compute cluster, which again could be an NVIDIA DGX box, can be something else, and you can open a Jupyter notebook or Jupyter Lab there, and from that notebook, in principle, you use Pyro client server modules to start experiment, right? So you can actually start the entire your entire experiment remotely, and then the data collected from in this experiment will be automatically saved on this network adjusted storage device, and all the data will be automatically available on that compute cluster, so you can immediately access it. Either you or whatever script you run can immediately access it from, let's say, from the Jupyter notebook, right? And then you do some analysis, and then the result of this analysis is again using the Pyro client server custom models, is sent back to a control computer, which decides what measurement to do next. So we don't decide. So the decision is made here on this compute cluster where you run your AI model, and or you can just do manual analysis. It doesn't really matter. And then the basically like something like a coordinate of the next measurement point in your parameter space can be sent to a control PC to do the measurement. But again, assuming, well, we don't have robots to to load samples, so you still need humans. But other than after that, in principle, like my colleagues, they are just they're running experiments from their home. So a lot of the AI driven or AI guided experiments that we are doing now, we just a person who, I'm not an electron microscopist, by the way, full disclosure, but people with whom I work, they just do it from, they run those experiments from their home. So yeah, so this is just a, but again, just so sort of this works for our specific environment, may not necessarily work for yours. And so just, I think that this general, framework, obviously you have your instrument, you have your external compute, and then you have some, network that allows you transferring information in between the two, right? And this the specifics of this network will be very different for different institutions. But and in a few years you probably won't need even this whole network thing, because you will just install a simple NVIDIA graphics card on your control computer, and it will just do all the job, because it will be more powerful than current supercomputers, just given how fast are the developments in that world. Yeah, so that's what I wanted briefly discuss, you can please check out a paper by Anis in eScience, and let me or him know if you have any questions. I think that's all. Yep.
Thanks, Maxim. So before I move on to the interactive part of today with the Colab notebooks, were there any questions on anything that I discussed or Maxim discussed? Don't be shy. All right. So let's then go to the interactive part. So as you probably already know, let me share the screen. So we're going to start with the July 5th ML course part 1 notebook. So you just click, if you're on Colab, you can click File, Open, or I guess I can just copy this link to you all and paste it. Copy. Here we go. So what we're going to do is first we're going to introduce the SidPy dataset object, and then we'll talk about PyCroscopy in the next notebook. But first, we just need to open this notebook and load the required packages. So there are a few things that are required here. I've already done this, but I'm just going to wait for you to do that. And once you run it, it will tell you to restart, but you don't have to do that. You can just click the next cell. And you should receive the SidPy version as being 0.12. So I just updated it a couple of days ago for the sake of this tutorial, because we've incorporated some new features. So I'm going to give 30 seconds until everybody gets to this step. You don't need GPUs for this. So if your runtime has GPUs, you can choose one which doesn't. So you don't have to use up your compute quota. All right. So the next step is just to check if we're in Google Colab or not. And that's just so that we can enable some interactive visualization. If you're in a notebook, like a Jupyter notebook, then you can use Matplotlib notebook instead. But this takes care of that automatically. So the first thing is we're going to just create a SidPy dataset object. So as I mentioned in the lecture, these are just sort of built on top of task arrays. And it's very easy to create one. You just need a single line, and it's say sidpy.dataset.fromArray. And so you provide a particular array and you give it a name. And so when we print this, what you see is that you have an array of this particular size, 4 by 5 by 10. So this is what we mean when we say n-dimensional. We can add another number here, for example. We can make it 6. And so now it'll be 4 dimensions. It'll be 4 by 5 by 10 by 6, right? So this is actually shown here, where this is printing you the data set itself. And it tells you that this is of data type unknown because you haven't given it a particular data type for the SidPy dataset object. It tells you the size, and it tells you also the dimensions that we have, right? So the first dimension is a generic dimension of size 4. The second one is a generic dimension of size 5. The third one is a size 10, and the fourth one is size 6. And so the power of the SIDPy dataset object really comes in specifying what these dimensions are, as well as what the SIDPy dataset type actually is. And so we'll go through that next. So, because this dataset is like a Dask array, we can do things like just slice it as normal. We can also do some interesting things, like we can reduce the size of it by slicing and return a SIDPy dataset object back, right? So datasets underscore cropped, for example, can be something like, because this is of size 4 by 5 by 10 by 6. So let's say 1 to 3, and then 2 to 4. So what happens when we do this, right? So this is sort of normal NumPy kind of slicing. But what you will see when we do this is that, in fact, I did not return a NumPy array. It actually returned this sort of, I returned this kind of array here, which is still, I think if it's type, we'll say that it's a SIDPy dataset object, right? So this is really useful when we want to take our data that's in a SIDPy dataset object and we want to crop it or slice it. It returns a SIDPy dataset back to us. One of the interesting things about or useful aspects about this SIDPy dataset object is, as I mentioned before, is that it allows us to store all the metadata, right? So users are really encouraged to capture the context regarding all of your data sets. So whenever you capture data, please add all the metadata necessary to describe it in the notes section or in some kind of form that can then be read. And so the SIDPy dataset object itself has these so-called required attributes. So it requires the quantity that's being measured. It requires units, right? So this could be the quantity might be, the response, right? This might be energy loss or something like that, right? So then it could have units. So obviously, if the response is in volts, or it could be an arbitrary unit or so forth. The data type itself, or what kind of data it is. So we have data types such as image stack, video, hyperspectral images, and so forth. And we also have sort of modality, right? So for example, it could be a photograph, it could be a TE, a micrograph, and so forth. And we also have the source, right? So we don't usually use this, but you can. And so this is typically used for things like what kind of instrument was it taken on, right? And so these attributes, if you don't provide them, are sort of set to generic initially. And so we can actually change the attributes for things like data type and quantity in units after we create the data set. So if you try something like spectrum image, this is not supported, but it tells you what is supported, right? So the types that are supported are spectrum, image, image map, image stack, image 4D, spectral image. So if this is a 3-dimensional data set, and so I'm going to go back here, I'm going to make it a 3-dimensional data set. Then what I can do here is, of course, this is not going to be supported, but however, spectral image is supported. And I'm going to say that, for the sake of argument, that the units are going to be nanoamps and the dataset quantity that we're measuring here is going to be current, right? Similarly, we can also save metadata as attributes. So these datasets can capture the sort of the rich scientific metadata that's associated with any data as attributes. And so one way to do that is, for example, let's say that we have some kind of calibration curve that is required to understand this data set. Well, I can simply set an attribute called calibration to these particular values, and then provide a dictionary within the metadata attribute within metadata, which gives me some information about things like, whatever I want, really, what is the phase of the moon, what you had for dinner last night, so forth. I mean, realistically, you'll be using things like what kind of microscope you used, what acceleration voltage, what sample was used. If you're on a user project, maybe what user project number, those sorts of things. But in any case, we can set all of these very easily. And so we can run this particular line. And so what you see here is that we can also print what was within here. So if you print what was in this calibration, then you see obviously this array of 0 to 4, which is exactly what was created here. And similarly, all the metadata that was stored as a dictionary can be read very easily and is stored within the SIDPy dataset object itself, right? So notice that you can't do this with NumPy arrays, right? So if you try this with the NumPy array, it's not going to work. But this really builds on top of sort of the existing Dask array package, so that we can enable all of these kinds of useful functionalities for scientific data. Has everybody gone up to here without any issues? Can you give me a thumbs up? Okay. I see a couple of thumbs up. All right, good. Excellent. Okay. So the most critical part for these data sets is the dimensionality information itself, right? So we know that, you know, in our original data set, we set these dimensions to be 4, 5, and 10 long, right? So, but what is how did these vary, right? So what are the parameters or what are the values of these particular spectroscopic dimensions? And so we can specify that within SIDPy dimension objects. And so we basically take our, let's go up here, sorry. We can set these dimensions, right? And so we just say the data underscore set, which is our SIDPy data set, and then we want to set the dimension. So the 0th dimension is going to be a SIDPy dimension object. And I'm going to say that, you know, at this point, it's just sort of a linear vector of, you know, basically the index of this particular size. We can give it a name. And in this case, I'm going to say that its name is X. Its units are going to be, in this case, micrometers. The quantity that we're specifying here is length, and the dimension type. So this is really important. These because we are considering that this is going to be an XYZ kind of or XY times some kind of spectroscopic dimension. The first 2 dimensions are going to be the so-called spatial dimensions. Okay. Are there any specific quantity values for metadata? I'm not sure what the question specifically means. But so quantity is typically the string in terms of what is it that we are actually measuring, right? So for example, in this case, the quantity here is the length that we are measuring. And so that's specific to each dimension that we have. So that's what we specify there. And the dimension type, as I mentioned, is spatial. And so we can do this here. The same thing here. We can set for the first dimension, except in this case, I'm providing some other values for the y-axis. In this case, we provided some values for the x-axis. Here we do it for the y-axis. And then for the third axis, we're just going to use some kind of, for example, sine wave, right? And say that, you know, the third axis is actually bias. So let's do this. And so we know that the dimension type for this one was spectral. We didn't mention the dimension type initially here, right? So if we didn't mention it here, then it's going to be generic. And so we can go back and we can edit those individual dimensions after we've created them if we want to. So this dimension type is going to be a spectral dimension. The units are going to be volts. It can be anything that you know, whatever you've captured. And the quantity is going to be bias, right? So let's do that. So let's have a look at what this looks like. So what does it say? So when we print out the dataset.bias, what does it say? Well, it says that bias is in volts and it's of this particular size. If we look at the first dimension, it says that this is the dimension y, which is named y, and it's a length in micrometers, and it's of this particular size. And similarly, this data set now is of type spectral image, and it has this particular size, and the data contains current, which is measured in nanoamps, right? And it also has this particular kind of metadata, right? So we have managed to make sure that everything that we need in order to represent this data is now within our SIDPy dataset object, right? So it's really, really straightforward. And what you can do now is that now that all of these things are defined, we can actually just use dot plot. And by clicking here, hopefully this works. There we go. This is an interactive visualizer, so we can have a look at our data and see what it looks like at different individual XY positions. this is very useful for a lot of the kinds of data that you'll acquire in STEM or in electron microscopy. Is that working for pretty well? Working. Okay, perfect. So the next step here is that the type of plot that you end up getting, right? In this case, this is an interactive visualization because it's a 3D kind of hyperspectral kind of data. And so it sort of splits the plot like this. But instead, if your data type was an image instead of a spectral image, then when you try to plot it, what it will plot is it will just plot your 2 dimensions, and then it will sort of average over the 3rd dimension to provide this particular 2D plot. So that's how that actually works. And so it really depends on what kind of data type that you specify. So make sure you specify this correctly. Otherwise, the kind of visualization that you get might not be correct for your particular case. All right. So we can also then take this data, which is right now in the SidPy dataset object, and we can push it to a file. So how do we push it to a file? The way that we do that is first we create a file. And so we're going to create these HDF5 files. And so I'm just going to give it a name, example file. And then I'm going to say, using H5Py, create this particular file. And so in order to create the file, after you create the file, we can save the results. This shouldn't say fitted, sorry. We should say save the data set with PyNsid. And so I'm going to create a group. And so if you know how HDF5 files work, they're kind of like directory structures where you take an individual file that has individual directories or groups, as they're called in HDF5 parlance. And so we can create these groups. And so within this particular group, I can write my NSID dataset, or in other words, my SidPy dataset object to this group, and I can give it a name. And so I'm going to just run this. We'll see how that goes. And then I can then close the file. So this then means that if I look here, I should be able to see this file. There you go, example file.hf5, right? So let's have a look at this file again. So this is how we can institute some reproducibility within our workflows. Once we have our data, once we have captured it in this way, and once we have saved them, saved it into a SidPy dataset object, we can then once we have imported it into a SidPy dataset object, we can then save it to a HDF5 file. And so what I'm going to do here is I'm going to obviously read this file again. And so again, we just use H5Py to open the HDF5 file. And then what I can do is I can have a look at what's going on here. And so we have a very nice utility called HDFutils. And so it has a bunch of useful functions, including print tree. And so what this allows you to do is to look at the directory structure or the group structure of any H5 file. And so when I run this, you'll see that in fact, this file that we had, it has this measurement and channel groups that we created up here. So if we go up here, we can see that we created this here. And we also have the data that we created. In this case, it's called my_data. It contains the dimension of bias X and Y. It also contains metadata pertaining to the instrument, as well as other metadata, as well as the metadata dictionary that we supplied it. And it also has, of course, the raw data itself, right? It's in my data. So if we want to read this back as a SidPy dataset object, we can use sci-fi readers. And so the way that we do that is we can instantiate the so-called NSID reader in sci-fi readers with this HDF5 file. And then we can say datasets is the NSID.read or NSID_reader.read. And so this is just going to provide us a list of all the datasets that it found, right? That were NSID datasets or SidPy datasets. So it found only one particular data set of note. And so I can look at this data, and then as usual, I can then go ahead and plot it just like I plotted it previously. So that shows you how you can read back in your SidPy datasets themselves from a saved file. And we can also have a look at the metadata that was acquired. And so as you saw here, there was a separate instrument metadata tag, and it has its own dictionary of metadata associated with that. So what I would like you to do now is please load your own data set using Sci-Fi readers, or you can create one in SydPy as shown up here at the top, and then plot your own data set using the .plot command and see if you can, for example, create a 4-dimensional data set. And the type of data that you should use is image_4D. So while you do this, I'll wait. I'll give you about 5 minutes. Or I guess I should give you a little bit longer than that. Maybe I'll give you 10 minutes. All right. Does anybody need more time? Or should I move on? All right, let's have a look at what I did. So what I did was I created a 4-dimensional data set. In this case, it's 4 by 5 by 32 by 32. And then I gave it its dimension information. The two X and Y dimensions were spatial, and then the two other dimensions were KX, KY. So these are spectral dimensions. And then, as I mentioned before, the data type here is going to be image underscore 4D. And so this one is supported. And so when I run this, I can get a 4-dimensional data set. And so I can go ahead and use an interactive visualizer to visualize what the 2D image is at each individual pixel, for example, right? So it's going to be a little bit slow because it takes some time in Colab to load all this. But this is sort of the advantage of using the SIDPy dataset object. It really allows you to do quick interactive visualization with really minimal inputs. And also gives you all the metadata associated with each of these. And so if you actually look here, for example, right, it gives you the units here, it gives you what the dimensions actually are. And so that's very useful when it comes to plotting. All right. So I guess the next step here is we're going to move on to the second part of the notebooks. So let's open the second notebook. I will paste it in the chat again. Let me just pull it up. Copy. Here we go. And as usual, we want to run the first cell to load our packages. So what we're going to do in this notebook is we're going to do some visualization and spectral processing with PyCroscopy. And so again, we use the SIDPy dataset objects here, but we're going to show you a typical workflow example where this could be very heavily used, or is routinely used, I should say. All right, so the first thing is let's download the required data, and we have some data here of different types that we can download. Okay, so let's check for Google Colab and then import our required packages and see how this goes. All right, so don't worry about this particular thing. You don't need Igor right now. So the first step is let's load and visualize our spectral data set that we're going to work on. We're going to load an IV data set. This is captured by a conductive atomic force microscopy. It's just an illustrative example. It's really not relevant exactly what it is, but it shows you the capabilities of the ecosystem. And this data was actually presented in this paper here in 2017. So what we're going to do is we're going to load the file. And so we showed an example of how to load HDF5 files before using sci-fi readers. We're going to read it into a SIDPy dataset object. And so we're going to hit play here. And so what we will get here is we will get an interactive visualizer where what I can do is I can plot the spectra at each individual pixel. This is actually a conductive atomic force microscopy data set of bismuth ferrite with spectra at each individual IV spectra or current voltage spectra at each individual pixel. So let's go, and so you can have a look at it, but it's plotted in an interesting way where I take the log of the current density and then the square root of the electric field. There's a particular reason for that, and that's just basically because if you're linear like this in this particular representation, you can then do linear fits here, and then you can this is actually kind of a Schottky emission kind of conduction mechanism. And so this representation shows the current to be linear should that mechanism be satisfied. All right. But generally what you do when you get data sets like this is that you would like to do some fits, right? So you'd like to see if I can do some functional fits on these individual spectra. And so this allows us to utilize the Dask array's innate parallelism. And so because we are fitting the same function across all of these different spectra, there's 2,500 spectra, but we fit the same function across all of these spectra. This is a very so-called embarrassingly parallelizable task, right? So what you can do is you can say that we have this block of computation, and I want to reproduce this computation across many different processes. And the good thing about PyCroscopy is that you don't need to know about any of that. All you need to know is the function that you want to fit and provide your data set. And so what we can do here is that we can provide the function that we want to fit. In this case, this is a linear function. So we're just going to define a very simple linear function that we want to fit. And then we want to instantiate the so-called SidPy class or SidFitter class. And so in order to instantiate this, we provide the SidPy dataset object, we provide the function fits, and then some parameters about what we want to use. So for example, if we want to use more than a certain number of workers that we have, if we have a computer with many different cores, for example, then we can use all of the cores. And so we can write the number of workers as being equal to the number of cores. If you have multi-threading available, you can use multiple threads. This is less relevant. I mean, you don't have to enter these. There will be some defaults here, but these work pretty well for Colab. And if you want to return a covariance matrix with these fits, you can say true. If you want to return the fit data set, in other words, return me the data set where the function parameters have been found, and then I use those function parameters to provide me the estimated fit for that particular spectra. If I want that data set back, I can say return fit is true. And then I can also use some more advanced prior determinations. So what I mean by this is that whenever we do a function fit, we have a prior for our function fit, which goes into the algorithm. And there are numerous ways to collect so-called better priors. And so that's described in this particular paper that we published in Machine Learning Science and Technology. You can read about it here. But this fitter class actually provides you the capability to incorporate the algorithm that's described in that paper. So anyway, I'm going to go ahead and hit play. And so what it's going to do here is it's going to set up the particular distributed computation, but it's not going to do any fit yet. In order to do that, to do my fit, I need to call the doFit method. And so with one line, we can now fit all of the spectra. So let's go ahead and do that and see how long it takes. It shouldn't take too long. So you can sort of see how long it's taking here. Probably should be done within 60 seconds. There we go, done. So 41 seconds it took for fitting 2,500 spectra, which is not bad, I guess. And so then now we have our fit parameters map. So the fit parameters are going to be A1 and A2. So see that here. So let's have a look. I can plot those here. I just do dot plot because this is also a SidPy dataset object. So this is just showing you those parameters here. And then the other thing here is that we can also do visualize our fit results, right? So because we have our fit results, what I can do is I can just call visualize fit results, and then I can just click here. And so what it will give me is it'll give me the fit as well as the raw data at each individual site. This is very useful for a lot of applications. So you can see that for the most part, the fit was very good. There were a couple of pixels where it didn't work very well, but all these other places, it actually worked pretty well. Maybe there's something going on at high voltages here as well that's not being captured by this linear fit. But for the most part, it looks really good. And then what we can do is we can then, as we did before, we can save the results. And so I'm just going to create a H5 file, create a measurement group, and then write this particular file, write this particular data set into here. I can look at the metadata. And so because we use the SIDPy dataset object in order to, and we use the SIDFitter class in order to fit it, not only does it provide me metadata on the individual data set, but it also provides me metadata on what processing occurred. So in this case, it says that this particular fitting function was used, and it just provides you the code of the particular fitting function that was actually used. So if you wanted to reproduce it, it would be straightforward to reproduce it by calling the SIDFitter object with these particular parameters and this particular function that is actually saved in the metadata itself. This is really useful for reproducibility. And again, I can have a look at what's in this particular file, and I can see that, I have my data set, and I have sort of the raw data set here, and then I have my dimensions that are associated with it. And so I can also put in my fit parameters and my fitted data sets here. So now I have my fit parameters as well as my fitted spectra that lie within the original group for the raw data set. So in this way, we have been able to load some data, fit the data, so do some processing, and leverage the visualization and parallel capabilities of PyCroscopy, and then save all of the data back, including the fitted data, back with its metadata into a HDF5 file. And so now we have a reproducible way to run this workflow. Are there any questions? All right, let's move to the second example then. So the second example is that we also have some sort of simple matrix and tensor factorization that's built into PyCroscopy. You don't need to run this matplotlib notebook here. We're just going to run this cell here. This is in case you are in a Jupyter notebook. So I have a DM3 file here. It's called bto_atomic.dm3. This is barium titanate. And so we can plot this data. So we're going to read this data using the DM3 reader, which is within SciFi readers. So I just say .plot, and you can sort of see this is the film of barium titanate on top of strontium ruthenate, and then strontium titanate here. So what we want is to only analyze the barium titanate portion. So how do we do that? Well, we can very easily crop it to only the film part. And so let's say something like this. Okay, that looks kind of nice. And so now you can sort of see some interesting structures within the barium titanate. So one way that we might want to analyze these images is using something called image windowing. And so what we do there is that we basically slide a window across this particular image. It's a sliding window transform, where we take image patches and then we do a process on those individual image patches. So typically that process might be something like a Fourier transform. It might be a radon transform. It might be some kind of image cleaning, like principal component analysis or so forth, right? But what we're going to do is first we're going to take these image patches. And then what we are going to do after that is that we are going to do we're going to apply that Fourier transform to each of these image patches. And then we are going to unmix that particular data set. So how do we do, so that we can see how the periodicity is varying across this particular sample, right? So if we do a Fourier transform of this image, you'll get some information about the overall periodic structure. But if we do a Fourier transform of each individual sub-image, and then we find out how that varies across the particular image itself, then what that gives us is it gives us more local knowledge about how the structure is varying, right? So in order to do that, all I need to do is I need to incorporate the image windowing or import the image windowing module from PyCroscopy. And it has some parameters that we need to supply. And so these parameters include what is the window step. In other words, when I move this window across this image, what step should I move in X and what step should I move in Y, as well as the size of the windows, whether I want to do a Fourier transform or not, and whether I want to use a window filter before when I apply this when I apply this Fourier transform. And so typically we use a Hamming or Hanning window, as well as whether we want to do things like zoom and interpolate. And typically this is quite useful because if you want to have a look at, pay attention to the individual reflections within the Fourier transform, it makes sense to often zoom in your image. And so I can call this module, and then I can basically make my windows. And then I can also do things like take an absolute value of the log of the windows, things like that. But then I can plot my windows. This will take a little bit of time because it has to actually create all of these windows here. And so this is going to create for us a 4-dimensional data set, right? At each individual XY position, I'm going to have its associated Fourier transform spectra. And so again, I'm just going to be able to plot it here. And so I can click here and I can find the Fourier transform at that particular individual pixel, right? It's kind of very useful. All right. So now that we have this 4-dimensional data set, the question is, so how do we analyze this further, right? So what are we looking at here? And a typical way that we do this is we use a matrix factorization approach. It's a very simple approach where we can do something like a non-negative matrix factorization, where we state that how can we reproduce the spectra with a minimum number of components? Let's say that I have a couple of sort of so-called n-member components. You can think of them as sort of basis functions. But how do we find these basis functions such that I can represent any of these spectra by a linear combination of those basis spectra themselves, right? So there are different algorithms used for this, including things like principal component analysis, independent component analysis, and non-negative matrix factorization. But typically for this particular case, we find that non-negative matrix factorization works quite well. And the reason for that is because it's based on the physics of the problem itself, where we think that, you can more or less represent these spectra as a linear combination where each component is obviously non-negative. You're not going to have negative components here of this absolute value of the Fourier transform. At the same time, we know that, we should have some sort of phase mixing, some sort of phase proportionality. So the linear assumption here is more or less mostly correct, I would say, or mostly justified. In any case, we can import the matrix factorization module from PyCroscopy, and then we can state what method we would like to use, as well as a number of components we would like to use here. And so I'm just going to state that we're going to use the non-negative matrix factorization, or NMF method, and then I'm just going to call the doFit module, doFit method within this module. So let's run that. And this is going to provide us the so-called abundance maps and components. In other words, the components are these sort of basis spectra that we can visualize. And then the abundances are how these basis spectra can be linearly added to each other with multiplied by certain coefficients. Those coefficients are the abundances of each particular spectra at that particular pixel location. In other words, if I want to represent this spectra as a function of, let's say, 2 other spectra, what are the coefficient maps? What is the coefficient of that first spectra and the coefficient of that second spectra that I need to multiply those basis spectra by to result in the best possible estimation of this particular spectra? So that's the problem that's being solved here. So if we run this, and I can then go ahead and plot it. Now, at this point, it's kind of difficult for us to be able to provide you abundances and components as SIDPy dataset objects themselves. And partly this is because of the dependent, it's largely dependent on the kind of methods that you use and the kind of input data set. And so we're still working on ways around that. But for now, you can just plot it using standard matplotlib. And so what we can do here is we can have a look at the individual spectra that were shown. So this is the kind of spectra that NMF came up with as the n members or the components, component 0 and component 1. And you can see how they sort of vary across the sample. And it's kind of interesting that they sort of form sort of banded kind of features here. And so this is potentially related to changes in oxygen vacancy concentration across and oxygen vacancy ordering, or semi ordering as it were, in this particular sample. And so we have a paper that discusses this in Advanced Materials. I think I don't know if I reference it up here. yes, it's the NMF is explained in this article, but there is a paper that you can search for barium tight and oxygen vacancies. If you put our names in, you will find an explanation of what we're actually observing here. All right. So I'm going to leave you to handle that for a couple of minutes, and then I'm happy to answer any questions you have. You can pop them in the chat as well if you don't want to unmute your mic. We can also change the method. For example, we can do ICA, for instance. This is independent component analysis, and then we can run this. So go ahead and see if you can try this on your own kind of image data sets that you have captured or you have access to. It can be quite a useful tool. So one question was, if we use the plotting and data fitting using PyCroscopy, is the data processing done at our computer or in the cloud? So right now, if you're doing it in Colab, then it is occurring in the cloud. If you're doing it in a Jupyter notebook on your computer, then you do it in your computer. But there's really no reason why the processing couldn't happen anywhere. You know, wherever the notebook is running is where that processing is happening. So that depends where your notebook server is running, I should say. Not your notebook itself, but your notebook server. That's the key point, because we can set up Jupyter servers on even HPC resources. For example, in OLCF, there's a service that allows you to run Jupyter notebooks on Summit nodes. And this is also true on other high-performance computing platforms. And it's obviously also true on the cloud, which is what we're running in right now. So, yeah. So you can see that if I chose independent component analysis, then I get some non-physically meaningless spectra because they have sort of negative values for these components. But I see some interesting kind of features in the abundance maps. But you can play with that and see how it varies when you change the number of components, for example. Are there any other questions? Is it working for everyone? Does it make sense? Okay, I see another question here. Is it possible to see the fit quality with the two components, and would this make it possible to localize defects? So yes, it is. So the way that you would do that is that you would have to have a look at basically combine the components together in using matrix multiplication with the abundances to be able to find each individual spectra itself, and you can come up with like a mean square error map. This would it be useful for localizing defects? Potentially. It depends on the defect concentration, because if the defect concentration is large enough, then I suppose that what would happen is that they would actually just show up in the components themselves, not necessarily in the error maps, because that means that it was not incorporated within the components. So that sort of it's dependent on that. But yes, you can easily just multiply the components by the abundances to reproduce the spectra and then see how well it fits. Yep. Thanks, Julian, for the question. Right, then I guess if there are no further questions, I'll leave you to play with this for the next 15 minutes or so. And if you have any questions, you can always email me. You can always contact me on LinkedIn, for example. And I'm very happy to answer anything that you might want to know about PyCroscopy or machine learning and so forth. So look forward to seeing you in future lectures as well. All right. Thank you all.